Good morning, everyone. I'm here again. My last lecture, we talk about the history of microbiology, and we end up with a little application of microbiology, especially the modern microbiology, where we talk about the use of modern microbiology or biotechnology applications. We stop there, but just to continue from the same, before we enter into the new topic of microbial diversity. So as far as the microbiology applications are concerned, we have a lot of microbial pathogens which can be used to, these pathogens can be used in order to make as industry. The name is given as a biotechnology. Using the microbial world transform into the technology for the benefit of the society, leading to a new emerging area called biotechnology. So how microbiology from look into in 1500 and 1600 in the century, now in 21st century, how it evolved in order to form a new technology emerged from the field of microbiology or the field of microbes, which includes all viruses, bacteria, fungus, and all. Well, so now we start a new topic today. The microbial diversity and systematic classification and modern methods and concept. Dear students, this is basically a very important area because until we learn the microbial diversity and the classification system in modern method, it is very difficult for us to understand the microbial world. The millions and trillions of microorganisms all across, around us. How do you name them? How do you classify them? How do you identify them? These are some major issues or major basically questions which need to be addressed. So in order to understand the microbial diversity and systematic classification, the importance of this topic is that to know which microorganism is characterized with certain features causing some disease or benefit for fermentation, for wine productions, for bread productions like fungus, Saccharomyces cerevisiae, for example. Are they harmful or useful? Or how can we engineer these microorganisms, all right, in order to make them useful? Then the concept of microbiota or microbiome is very important because whole microbiome, gut microbiome, oral microbiome, or any microbiome, even mud microbiome, soil microbiome, environmental microbiome could have some significance. So in order to understand what is this microbiome is total made of, what are the different microbial flora, microorganisms present in this microbiome, can be used through several techniques. So we call modern methods, some traditional methods, some new concepts in order to learn. So we'll learn in this topic, we'll learn all these things. Well, the vast world of microbial diversity. Diversity in the bacterial population, diversity in the fungus population, diversity in the virus, diversity in protozoan, diversity in algae. So, within the domain of each, we have a lot of variations, even single species. For example, if you talk about the E. coli may have hundreds and thousands of different strains. How these strains are diversified, how these strains are different. So these things we'll study it. Suppose you pick up the E. coli strain from clinical isolate or from environmental sample, then this E. coli may be different from the other E. coli, although both are Escherichia coli. Similarly, Klebsiella pneumoniae or Pseudomonas aeruginosa. So each of these species, beyond the species, we have a lot of variation also. So see the level of variations, and to comprehend the whole and to understand the whole diversity, microbial diversity is very, very important. So what are the tools to understand? What are the parameters to learn it and to identify these is very important. Now the microbes are invisible majority. Now 60% of Earth microbiome or the biomass, right, is basically constitute of the majority of the living matter on our planet, demonstrating their fundamental importance to global ecosystem. Then prokaryotic cells, the estimated number of the prokaryotic cells globally representing an almost incomparable abundance of the microbial life. There's a lot of different microorganisms are being present. Then 99.9% almost undiscovered microorganisms. Undiscovered microorganisms means like microorganisms which are not known, not aware about it. They cannot culture. So the problem with this, we have majority of the microorganisms which are basically non-culturable, which are hidden, which are not aware about it, we are not aware about it. So how to understand these microorganisms is through new technologies. We'll discuss this. Why study the microbial diversity? As I have told you earlier, the microbial diversity, to study the microbial diversity is important because until unless we understand their diversity and their each and every species and genera, phyla, class, right, to know what about it, we cannot work on it, we cannot learn what these microorganisms are doing. So medical breakthroughs. In medical breakthroughs, the microbes serve as a primarily source of antibiotics, life-saving drugs like penicillin, ampicillin, and have revolutionized the modern medicine from penicillin to modern broad-spectrum antibiotics. Now, the microbial diversity continues to provide new therapeutics and compounds for treating the resistant infection and emerging diseases. So, this is the medical breakthrough. So, there is a category of microorganisms which are basically causing the disease as well as preventing the diseases through production of the various antibiotics or antimicrobial agents or compounds. The industrial application of the microorganisms also. The microbial enzymes powers numerous industrial processes from the food production to textile manufacturing, and these biological catalysts offer the environmentally friendly alternative to harsh chemical processes, enabling sustainable manufacturing practices across diverse industries. Well, sustainable energy, the microorganisms hold the key to next generation biofuels. This is the next generation biofuels, which is one of the major challenge before us, offering the renewable alternative fossil fuels through the possible like fermentation and biogas production. Microbial diversity enable clean energy solutions and that reduce our carbon footprint. Environmental solution is also one of the area. The bioremediation harness microbial capabilities to clean up environment pollutant from oil spill to heavy metals contamination. So you can engineer some microorganisms. You can identify certain microorganisms which can clean the spill, oil spill, which can degrade the oil on the sea or on the ocean or any water bodies. And these natural processes offering cost-effective and environmentally sound approaches to restoring the damaged ecosystem. Now, these microbial flora or microbial world is basically help us to understand how we can control all these things. The microbial world, the incredible diversity of microbial life, including bacteria, viruses, fungus and all. From simple bacteria to complex archaea, from beneficial fungi to intricate viruses, each representing unique evolutionary solution to the life challenging, right? How? The classical method and the modern method. So first we'll talk about the classical method in microbial systematic classification. Now before we go that, let me clear you, there's the word classification, identification and characterization. So the classification means you classify the bacteria, viruses, fungi in different classes, phyla, order and different hierarchies. Now identification. What are the basis of identification of these microorganisms to be classified into different group, class, and all, and then to characterize it. So all these three things, classification, identification, characterization, are hand in hand, and characteristic features are identified, right? Or we call some manual characteristic features are identified, which help these microorganisms to identify and to keep it into certain class, order, phyla, kingdom, and so on. For example, whether the microorganisms or the living organisms belong from animals or plant or microbial world, right? Are among the microorganisms from bacteria, virus, fungi, protozoa, algae, fungus, and so on. So all these kind of a classification based on certain characteristic features, and these characteristic features to be identified. So classical methods help us to identify these characteristic features. For example, morphology and cultivation. The early foundation in early microbial foundations, microbial world foundations, the simple morphology is one of the major parameters. How we can look into the morphology, whether the bacteria are the spherical, bacteria are the elongated, they are spiral, they are different shape, different colors, right? So all these morphology and colony shape, colony structures, textures can be identified, and based on these simple visual screening of these microorganisms, right? Looking at the colony size, looking at the color, looking at the texture, will help us to categorize the microorganisms into different categories, different classes, order, phyla, based on these unique characteristic features. Now, then come to the nutritional requirement. So what are the nutritional requirement? Nutritional requirement means different microorganisms have a different nutrition, right? Based on their nutrition value, based on their nutrition requirement, based on their metabolic processes which metabolize different nutrition. For example, some of the microorganisms, they grow on the carbon sources like a dextron, sugar, glucose. Some of the microorganisms, they grow in the presence of salt. Some of the microorganisms, they ferment the fructose, some sucrose, some glucose. So basically, depending upon the metabolic processes, metabolic setup of microorganisms, they utilize the nutritions. So indirectly, nutritional requirement is also one of the indication of the microorganisms to be categorized into different categories. Then pure culture isolation. So culture characteristics is again, now in case of culture characteristics, you have to know what are the different culture characteristics. Again, these things are quite related, because if you have a different nutrition, different carbon source, different energy sources to provide the microorganisms, so for example, bacteria need a different media. Bacteria need a different culture conditions. Virus need different culture conditions. Fungus need different culture condition. So having a different culture conditions, you can have identified different microorganisms to be categorized into different order, class, and different hierarchies. Now, the phenotypic classification challenges has major limitations. For example, morphological convergence. The similar environmental pressures often produce a similar cellular form across the unrelated lineages. So in certain cases, these microorganisms morphologically same. So sometimes it is very difficult for you to categorize them into one group or to other group, but they are same. For example, if you talk about the coccus, spherical. All these spherical microorganisms, they are on the same category, so you keep them into one class or one group. But the other microorganisms, which are also spherical, how to keep them into the same group or different group is a big challenge. So these are some limitation of the morphology. So you should have a number of the different characters involved during these morphological coverage. Phenotypic plasticity. What does it mean? So many microbial traits, right, proved highly variable depending on the environmental conditions. For example, some of the microorganisms, they have adapt different conditions, and having adapted different conditions, they can change. So they make a confusion whether you keep it into one category or into another category. So making consistent classification extremely difficult for scientists to keep them into one class, group, or one hierarchical status. Limited resolutions, phenotypic approaches couldn't distinguish between closely related species. or identify cryptic diversity within the morphological similar groups. So these are some challenges with the morphological or phenotypic conditions. Now, historical milestones during the classifications, the Lenin system of classification adapted for the microbes were the historical benchmark. In 1753, Lenin Foundations initiated, that is Carl Lenin, established the binomial or binomial nomenclature. So it means the nomenclature of these microorganisms by giving two names. Bio means two names, nomenclatures. Now the plant and animals providing the hierarchical framework that would later be adapted for microbial classification. Okay. In 1872, bacterial classification starts. Now the Funin and Kohn, the first applied systematic classification of bacteria, he was a scientist, establishing genus and species. So the identification or giving the nomenclature of a genus of a species, genus and species of an individual is started in 1872 by Funin and Kohn. What does it mean? It means, for example, Escherichia coli. So Escherichia is a genus, coli is a species. Now, under the genus Escherichia, you can have a number of different species by found. For example, Pseudomonas aeruginosa, Pseudomonas putida, and there are many more. So, under the genus of a Pseudomonas, you can have a different species. So, you have to identify some characters which are related to the common genus, and then unrelated characters in the species. So, establishing the genus and species designation, given based on the several morphological characteristic features. Then in 1923, the very recent in history of classification, the Buggy's manual was standard benchmark for the classification. The first edition of Buggy's manual, Determinative Bacteriology, provides this standardized classification criteria through still based primarily on the phenotypic trait. Now, these are all conventional method of classification where the phenotypic expression and morphological and cultural and nutritional and metabolic conditions are to be considered into characters identification. In 1980s, a molecular revolution came, and in molecular revolution, even the species beyond a species can also be identified. Now, the integration of the molecular techniques began transforming the microbial systematic, revealing the inadequacies of the purely morphological approaches. And these morphological approaches are basically help us to understand what are the basic criteria for this. Now, the visible tip of the microbial iceberg, the classification cultivation methods revealed only the fraction of a microbial diversity, right? Like seeing merely the tip of an enormous iceberg. So, whereas the 99% and the majority of the microorganisms are invisible. They cannot be cultured, they cannot be seen, they cannot be identifying. So, to explore them is a big challenge, right? The molecular revolution in the microbial diversity reveal a new area where you can understand what are the molecular methods and molecular tools which can help us to identify exactly the microorganisms with the unique characters. Now, as we have discussed earlier, that the conventional method, which are based on the cultural characteristics, which are based on the the nutritional value, which are based on the metabolic responses, which are based on the staining, which are based on several other. These are all methods which are based indirectly where you can use the phenotypic expression. Now, what is the phenotypic expression? Phenotypic expression means the expression of the individuals, bacteria, virus, fungus, and other, in order to show their characters related to individual microorganisms as a unique characters. So what the scientists do, they enumerate these characters 1, 2, 3, 4, 5, 6 based on different morphological, right, different cultural, different nutritional requirement, and so on and so forth. And then they compare the other microorganisms with again enumerating the 1, 2, 3, 4, 5, 6 characters, and then they compare these two. And they're identifying these two microorganisms based on the percent similarity and percent differences. Now, percent similarity and percent difference is basically help you to differentiate these two based on certain benchmark, based on certain criteria. Okay, this much percent differences, percent dissimilarity between these two microorganisms, for example, A and B. So we can keep A as a different group, B in different group. Similarly, if the percent similarity is reduced, so we can keep reducing the group and we can bring them into the closed group. But these morphological characteristics through intuitive method or through some other method, they may not be very reliable to exactly identifying the microorganisms based on the characteristic features, morphological characteristic features, and to keep them into one classification. Now, what are the requirement of the classification is basically the reproducibility, predictability, right? What does the predictability means? Stability and predictability means the characters which you identify for one microorganism should be predictive in terms of, like, if other microorganisms being identifying from clinical isolates, from environmental isolates, from any water sewage, water baselines and water bodies, then these organisms, if show similarity, percent similarity to these organisms, earlier one, then we can look into the percent similarity characters. If the percent similarity characters are same and they are predictable, for example, say for example, color of the colonies. If the color of the colonies is a pinkish with a luster color, then you say, okay, it is E. coli on certain media. Now, if other organisms identify is also close related to the same color and texture of the colony, it means you can predict, yes, these organisms may be of the same kind. So these are the conventional methods which are taken into consideration in order to identifying the microorganisms and keeping the microorganisms into particular group and order. Now, these methods are basically not very reliable. So in order to understand the exact microorganisms based on the molecular characteristic features to be kept in one individual organism so that it may not be repeated again as a wrong organisms to be kept in the same group, has to be taken into consideration the molecular characteristic features. Now, what are these molecular characteristic features? So these molecular characteristic features identifying through different molecular techniques. So we call the molecular evolution in the microbial diversity. Now, the detail of this topic on molecular evolutions in the molecular diversity will be discussed in my next talk, but just I can give you a brief introduction about it. We can have a DNA-based molecular techniques where we understand the genotype. So there's the two things, one is phenotype and there is a genotype. So we'll talk about the genotype in the next lectures in the detail, and then we'll discuss how the genotypic characteristic features will very precisely identifying these microorganisms from one to other. Thank you very much. We'll see you in the next